Okay, well, once again, on behalf of Coffee with EMBL and Thomas and Nushnai and the team, welcome to everyone. If you're here for the first time, great to see you. For everyone else, welcome back. We're celebrating something of a milestone today, I think, because this will be our 30th episode of Coffee with EMBL, and I think that's quite remarkable. I certainly had no concept when Noosh first suggested the idea of having a Coffee with EMBL session that there would be more than one, let alone 30 of them. So I think the fact that we've kept this going and used it as a forum to bring the community together is remarkable. And we were just chatting earlier about how nice it has been to be able to link up with our friends and former colleagues from around the world, really, and that people from so many different countries now can participate. And we will try to find out as well. Perhaps you can check, Rory or Tom, into the statistics of just how many countries we've had participants joining from. But I think it's a good thing to just, point out and remind people currently working at EMBL just how big a reach EMBL has and how far the alumni extend, as well as the current staff. And it's great that this has become an event not only for alumni, but also for current staff of EMBL, who are all, as we like to say, alumni in waiting, in training to become alumni. Once again, just to remind everybody that we do record these sessions. We realize that due to time differences and scheduling conflicts, not everyone who would like to participate is free when we do it live. But we do create video transcripts and put these online. So please do go check them out, tell people about them. And if any of the previous events you'd like to see, they're, I believe, all available on the website, and you'll be able to find it, or they'll be able to put links also here in the chat for that. We do encourage everyone to participate. So please do post comments or let us know if there's something you'd like to say. And we have a set of people, as usual, to lead the discussion today, but feel free, everyone, to contribute. We're having the theme this year, in the 50th celebratory anniversary year of EMBL, of just reviewing EMBL across its first 5 decades of existence. And today we've made it as far as the noughties, so the first 10 years of the current millennium. And we're obviously looking forward to many more to come, maybe even more millennia, I don't know. But we've got a great lineup of participants and discussants today. So I think, with no further ado, we'll start off with Eric Karsenti, if that's okay. And Eric, of course, was at EMBL for quite some time, including during the noughties, and has had a wide and varied career. Eric, I was reading some of the comments you forwarded, and just in starting off, before asking you to give us some thoughts about your time at EMBL, something that really struck me about the comments you had written down and discussed is that you felt when you were at EMBL that everything was possible. So I hope you remember writing that. But I think that's really such a great way of capturing what EMBL has meant to many of us. I felt the same. And maybe you could just tell us why it felt like that.
Yes, well, when I came, I started to work on very different things. I mean, on the cell cycle, on morphogenesis in cells, and things like this. And at this time, it was not like now. I mean, we did not have much information about the molecules or how everything worked. And each time I started to do something very far out, I mean, I found out somebody who could help, or come up with ideas or with tools or everything. And almost everything I wanted to do, I managed to do. I was very surprised because, in many places, when you want to do something, you have to ask for people, but people are reluctant, or there is a competitive aspect or something like this. Here, it was very collaborative, but in addition, the technology was amazing. I mean, it was just always at the top. That you could have everything you want to have to do the work. So this was fantastic. Yeah.
And something else I'll just mention or remind people that thanks to Noosh bringing us together with this forum, together with Noosh Thomas and I had this idea that we could also develop some one-to-one interviews and put them online in a YouTube channel. So we've started doing this, and we've already recorded a number of interviews with EMBL staff and alumni. So please go and check those out. Again, we can have the links to those videos, and there will be many more coming online soon. So Eric, you were one of the people I had the pleasure of interviewing this July at the occasion of the 50th anniversary celebration. And one of the things I remember being fascinated about in our discussion was your experience also with the Tara Oceans project. Perhaps you could share some thoughts on that with us.
Yeah, well, that was a long shot. I mean, I started I started to think about an expedition like this long before we started the expedition. I started to think about this in 95, I think. The idea by reading the book of Darwin, The Voyage of the Beagles, I thought it would be interesting to redo this. And the first project was actually called Evolution 2009. It was more about trying to redo the voyage of the Beagle, try to popularize cell biology, developmental biology, and evolution, obviously. But this did not work out, and finally it took a long time. And in 2007 or something like this, I realized I should, if I really wanted to do a proper expedition, I had to do a real scientific expedition and use it to popularize science a little bit, but the core should be science. So I found Tara and the people who were running Tara at the time, Romain Troublé and Étienne Bourgois. And I suggested them we could do something together, and that's how it started. So, but I spoke a lot to all the senior scientists. I was bothering everybody with this for many years, before we started the expedition. But I did not really think it would work one day. I mean, but finally it has worked out, and it's an amazing project now. It is continuing at EMBL with the trek and around the Europe expedition with Tara. And I think this is very important because we discovered a lot of genes, and also it's a completely new way of doing marine biology because there is a tight connection between biodiversity at all scales, from viruses to zooplankton. And in addition, we collect a lot of environmental parameters all over the world. So this is very nice. Yeah, it's a great.
Where can people find out more information about that project?
I think there is a site at EMBL, but there is also Tara Oceans. I mean, now the Tara boat. I mean, the people who run the boat have created the foundation, which is called Tara Ocean without an S, but Tara Ocean. And if you look on the web, you can find a lot of stuff on there. I mean, it's plenty of information.
Yeah. I think this was a fantastic project for communicating the excitement of science to a wider community and also to young people to interest them. I can share one very short anecdote. When I left EMBL and became an alumnus and moved back to the UK, I was invited to participate quite early on with an event that was organized with TV representatives to talk with scientists around the UK with a view of thinking about things that might interest the general public. And whenever I spoke to them and said I was interested in cell biology, they all said, "Oh, the public don't care about cell biology. There's no interest." So I'm very impressed that you found a way that you could make cell biology interesting. I wonder also in your role at EMBL and during the noughties, during the first decade, you took over from Kai Siemens as head of cell biology. And what was that something that you had in the front of your mind, the idea of not only doing great science, but also communicating with the European public?
Well, not really. When I started the cell biology unit, I was still very excited about just trying to understand biology, I mean, cell biology. And we knew so little about, we started to know a little bit about molecules and things like this, but we didn't understand much about, cell organization and cell dynamics and all that stuff. So I was really completely excited about the science at the time. I thought it would be important to get the general public interested, but I did not really think of doing anything uh, really about popularization at this time. Yeah, it came later, but not at the very beginning when I started to take over from Kai. Actually, this was a huge of work, a huge amount of work just to start this new program. Yeah.
Yeah, I can imagine. Thomas, do you want to follow up?
Yeah, well, I mean, in thinking about this part, I was thinking also that cell biology was also famous for the high level of parties and social life, and I think when the big move happened to Dresden, there was a lot of people, Kai and Marino, who left, and so I was wondering, I mean, how was your contribution to keep up the social challenge at the
I did
EMBL
not.
in those days?
Well, unfortunately, I did not do much about this. I mean, it was Kai. Kai was really Kai and Gareth. I mean, Gareth Griffiths and Kai Simmons were very active in running this. No, I kept doing this for the group leaders. We had retreats all the time, where every year, every 6 months. I don't remember exactly, but at least every year. And during the retreats, obviously, we had we had a lot of fun. But the thing that Kai was doing, like the living parties, for example, I stopped doing this. Yeah, I didn't have the energy.
So it ended up being all on the shoulders of Garrett.
Yeah.
All right. No, but I think this was the time when I joined EMBL, and it was a big time of change. And here we have with us also Nadia Rosenthal, who was joining around that time. And I think one of the things that I remember was that Nadia came around that time with a big task ahead of her. So, but before we talk about that, so I was wondering what brought you to the MBL, and what attracted you. I mean, you were coming in from the States, if I recollect, right?
Yes, I was. I was first approached by Fiscofatos when they were just starting the EMBL unit down in Rome at Monterotondo. And I remember at the time, he says, "Oh, you should go and run it." And, it was obviously a little bit of a move from Harvard Medical School. So I called Peter Gruss, who at that point was about to become the president of Max Planck. And I had been a postdoc with Peter at NIH. And I said, "Peter, what should I do?" He says, "No, don't go unless you really want to understand how to make good pasta." So I said, "Okay." And then, so then Klaus Rajewski accepted the job. But then Klaus got all wound up in going to Harvard himself. And so Fotis calls me back and says, "Look, why don't you reconsider?" So of course I call Peter. By this time he's the president. And I said, "Okay, now what do I do?" He says, "Now it's good. Push the button." So that's all, it's all Peter's fault, but not really. And the main incentive for going was two people, very important people in my life. One was Anna Frusi, who was my best friend since we were graduate students together at Harvard. And I had kept in touch with her, and we kept saying, "I miss you, I miss you." And so she said, "No, come on, take this job. You won't regret it. EMBL is amazing. You've got to take this job." And at the same time, she had introduced me to my husband, Alan Sawyer. Alan was working with Eric, and I can tell you that Eric is being very modest about the parties, because indeed, all I hear about were parties. So in any case, Alan said, "Oh, I'm really kind of sick of the US. Let's go back to Europe." So one thing led to another, and I ended up in Rome. But one thing I want to say before we get on to the topic of Rome is going back to Eric and Tara Oceans. Eric, I want you to remember the first thing that we tried, which was we went to the Royal Society. I don't know if you remember this. And we had this hilarious interview with Russ Hodge and you and me, because we had this vision, to do this, the Voyage of the Beagle. And I was totally excited because I'm also a sailor. So the three of us went, and it was one of these huge rooms, you know, one of those incredible rooms in London, which are scary. And there were all sorts of scary old people sitting around, looking extremely intimidating. And furthermore, there were two interviewers who were going to interrogate us about this project, one of whom was Richard Dawkins. You know, no pressure, no intimidation there. And the other one was a TV producer who I remember was wearing a blue velour suit with a great big orange marigold. He was quite strange and extremely obnoxious. And so we told our story, and Richard said, "Oh, this is going to be fantastic. You should do this. Absolutely." And then this TV guy says, "Oh, this is terrible. You need to turn Darwin into a young boy, and then you need to make it into an action movie." And, you know, by that time, Eric was like, "Oh, no, science. So in any case, we didn't get the money, but Eric didn't stop there. And that's how Terra Oceans then
but
became.
You forgot to say something that was even funnier with this guy. He told me, "But you are French. Why do you want to do something on Darwin?" So for me, this was really amazing because, you know, I had completely forgotten about nations. It was 25 years I was working at Temple. I was completely European somehow.
Right.
So this was a completely knocked down question. I could not answer to this one.
Yeah, I think this highlights the idea of community at EMBL that really transcends nationalities.
Yeah.
And Nadia, you commented on the scientific community and the community of the EMBL as being one of the reasons you came to the EMBL, right?
I mean, it was just so exciting to be able to start to attract people from all over Europe. And indeed, we have one here, Paschalis Kratsios, who I'm sure you'll have a chance to meet and talk to, who is one of my top star productions. I have to say, this guy is unbelievable. And I'll never forget when he came to interview. He came from a rather small university looking for graduate work. And he had grown up in Greece. And he came in with about 4 crazy ideas, but were all related to things that I was already working on. So the guy had read all my papers and was already being creative before he even got, I mean, you know, it was obvious I was going to take him. And since then, I mean, just one thing after another. I know I'm talking like a mother, but it really, he went from being interested in the mouse to being interested in neuroscience, to going to Columbia to use worms to study this. And now he's a big shot at University of Chicago, just north of where I am right now. And this is just one of many wonderful stories that come out of these extraordinary groups that form. I have to say that Montreal, it was very exciting at the time, because everybody who came down from Heidelberg said, "Oh yeah, this is the way it used to feel at EMBL when everything was muddy and there were terrible."
I actually wanted to ask you about that, and maybe we can bring in also Paschalis on these. I mean, because you say, you know, it's exciting, it's a great place, but I think, you know, also being Italian, it must have been quite you know, a challenge to reshape what was there. So what was the challenge? I mean, how did you think of you know, as a direction for the unit? And maybe we can ask you, and then maybe get also the point of view of Paschalis.
Well, so, you know, my idea was we're all going to be working on the mouse because it was the mouse biology unit. And so what we should do is bring in as many different ideas as possible about how to approach biology from the mouse perspective, and not to be too prescriptive about this, because we were right in the middle of a period of great excitement about mouse knockout. I don't know if you remember, but at the time there was an extraordinary blossoming of excitement having to do a lot with Cre recombinase and the capacity to make conditional mutants, but also the idea that we could manipulate genomes on a much grander scale. And so with this came my idea, which is don't prescribe what tissue, what system. Let people find an interesting gene and it will take them all over the place, because you knock out a gene, you think it's expressed in the kidney, you think the kidney's going to fall apart, the kidney's fine, the heart falls apart. And then you have to find somebody who knows something about the heart. So if we have at least some of this knowledge inside of the institution, and of course, being able to then talk to people in Hamburg, in Grenoble, in Heidelberg, EBI, we will find out more about biology and the connections between tissues and cells than we will just simply hammering into our own little spot. And I think it was quite successful, but Paschalis should really comment on this.
Yeah, so Paschalis, tell us what was, taking part of that endeavor.
Yeah, absolutely. So first of all, hello, everybody. It is a great honor to be in the same chat with my scientific heroes, and especially Nadia, who is responsible for the scientist I am today. So I just want to say that, yes, it was really an exciting time for mouse genetics, and it was a time before CRISPR-Cas9 gene editing, right? So developing a mouse mutant was the main challenge was how much time it will take back then, right? Right now you can do that much faster, but nevertheless, having 6 or 7 groups at Monterotondo, international groups working on mouse genetics and reverse genetics was something that really opened up my mind as a student. And as Nadia said, I fell in love with genetics, and then I went from reverse genetics, where you just take a gene one at a time and you mutate it, to forward genetics. So I was still in the genetics field, but I went in another organism, in the worm C. elegans, right? Where I wanted to do unbiased genetic screens and figure out how the nervous system develops. And up to this day, 20 years later, so I joined the EMBL when I was 22. And 20 years later, I still have mouse genetics in my lab, combined with C. elegans worm genetics to basically tackle questions related to nervous system development and disease. I just want to say a few things from the perspective of a young student, if you don't mind,
Sure.
Thomas, because I just want to highlight what an opportunity was for me to join EMBL. So it basically broadened my horizons. It was the second time I got into a plane in my life, and that was to EMBL Rome. And these were formative years for me. And what I remember was the scientific environment at the EMBL Monterotondo, which was very international. We had students, postdocs, PIs from all over the world and Europe. And also the trips to Heidelberg, the frequent trips to Heidelberg during the Predoc course, where I still remember the lectures from Eric Karsenti on microtubules and cell biology. But I just want to say that it was a really international environment, where students could be mentored by international PIs or postdocs. In Nadia's lab, I'm very grateful to be under the wing of Fotini Mulchioti, who is also an associate professor at the University of Pennsylvania now. And I know that the time is precious. I want to highlight 2 things. Coming from another country and living in Italy, as you mentioned earlier, for me it was an exciting time. It was a time of continuous discoveries inside and outside the lab, right? Inside the lab, we were trying to figure out how the heart develops, how the heart functions in Nadia's lab. But then outside of the lab, I had to learn a new language. I had to get to know the Italian culture, the Italian people, and I fell in love with that. Up to this day, I cannot start my day with the best quality espresso I can have, and after lunch, there is no way I'm gonna order a cappuccino, right? So these things have stayed with me forever, and I will be always grateful. So I think, that's all I want to say.
Yeah, no, but I think, being a predoc around that time, and I think for most of the people that have bloomed at the EMBL, I mean, I think the atmosphere and the community connected to together with great mentorship and the international environment, I mean, really empowers people. So I think these are the things that we all love about the EMBL. So thank you for your comments about that. So yeah,
I was just going to say I think that I really come to regard the EMBL Predoc program as the jewel in the crown of EMBL, and Thomas is very modest, but in fact he's also a wonderful example of graduating from the EMBL Predoc program. I was just interested to ask Paschalis how you heard about the program and what attracted you to switch countries and do that at a tender young age, to leave your home and go and do that.
Right. So we have to remember that 20 years ago, social media were not where they are today, right? I barely had an email account, right? So
you mean you went on TikTok at the time?
Right. So I heard about, obviously, when I was in Greece, I studied I got my bachelor's degree at the University of Democritus University at the border of Greece and Turkey. And there we had a lecturer, I still remember his name to this day, be prepared, Raphael Sandazopoulos. Okay, very Greek name, but he had spent time at EMBL. So he had planted a seed in my mind about EMBL. And then the opportunity came, I was so lucky. I sent one graduate school application, and that was to EMBL Monterotondo. And I was so lucky to be accepted. So that's how I heard. I knew from my lecturer in Greece back then that's a great place.
And Nadia, was it your experience there that you got a lot of interesting applications from very varied types of students wanting to come to Monterotondo?
Well, yes, and there's a fantastic and very romantic story, which I'll be very, very brief about. And this had to do with the fact that when Paschalis joined, I had also been very attracted to another extremely compelling PhD candidate. And her name was Katerina, but we already had two Katerinas in the lab, so we ended up calling her Catella, which was her last name, so she became Catella. And so you can't tell, but Paschalis is an extremely tall person, and Catella was shorter than I am, and I'm not very tall. And these two were fighting all the time. They were continuously having, I mean, not in a bad way, but academically they were fighting. They had serious, fierce arguments about science. And they were, I could hear them, no. She was from Portugal, she's from Greece. They're yelling, screaming in English or God knows what. And anyway, so just to make a long story short, I had many conversations with Paschalis. How you doing? How's your life? Are you acclimatized to Rome? I says, yeah, but my love life's in a shambles. So I said, oh, that's really sad. I'm really sorry to hear this. So then we were in, I didn't know if you remember this, but we were in Belgium sitting around drinking beer, and it was at the wedding of one of our other students. And Paschalis looks at me. Everybody on the table is, a long, long table in the middle of a square, beautiful square in Leuven. And Paschalis looks at me and says, well, he says, my romantic problems are over. I look at him. I say, oh, really? What's going on? And the whole table completely dissolved into laughter because, of course, he and Catella had fallen in love. And I was the last to realize it. And of course, now, how many kids? I can't keep track.
No, we have one son together.
Anyway, so and obviously this was one of the sweetest stories, but it's just typical of the way people find common activities and then common interests and then eventually really a common life by interacting with each other in this extraordinary kind of melting pot that you get at EMBL. I'm sure there are about 100 stories like this, but this one just touches my heart.
Yeah, thanks. And again, I think it was a great thing that during the noughties we saw the EMBL organization expand and set up additional laboratories that could spread the mission for training and integrating people. I think that's been a fantastic success story for the whole mission of EMBL, and I really hope that it's something we can build on. And at this point, I'd quite like to bring in Allyson and Jaak from the EBI. So, you know, in our previous Coffee with EMBL, we talked about how what at the time was the data library, which and all the colleagues that we had in Heidelberg then made this transition, and I think literally packed up some tapes in the back of a car, and maybe we can hear about this, and drove them across the channel to Hingston, where the EMBL-EBI was being established, and so I guess we've moved on then, and now we're EMBL-EBI has been established for a short number of years and is growing rapidly. Allyson, how was your experience in working at EBI in the noughties?
Thank you, and thank you for having me, everyone. Yeah, I told you about how Peter Sturr had told me how he had driven those tapes, those hard disks that had EMBL on them all the way to Hingston. It boggled my mind at that time, even in the early, 99, 2000. Now that I've been thinking about Peter Sturr, I've suddenly forgotten the actual question you gave me. Sorry about that.
Well, no, I was really just wanting you to tell us about your experience of working at EBI in the noughties, and in the next decade. EBI had, I guess, gone through the transition of establishing itself and was growing, and you were developing this amazing role providing services in bioinformatics across Europe.
Absolutely. It was my first real job out of my master's degree, and I went straight from a master's degree into the EBI. And at the same time, Paul Kersey joined, and Tom Owen joined, and Jaak, I think, was the same year as me. It was the first big expansion. There wasn't the extra buildings that there is now on there. It was just the original EBI building. I didn't realize when I joined just how much it was growing at the time, but it really opened my mind to what science was about. And I think that all of our previous speakers have been talking about communication and engagement and community. And I think more than anything, the EBI made me understand what it meant to be part of the scientific community. And I don't think I could thank them more. I don't think it'd be possible for me to think of, I could have any better introduction to my scientific career than working at the EBI and learning how science was as much about community as it was about research. And I was in Rolf Weiler's team, which with Trimble and SwissProt, which ultimately became Uniprot, and my bosses were Claire and Maria Jesus, and it was just the most fantastic place to begin that journey.
And Jaak, did you also have a similar experience in working then at EBI? Oh, you're muted, Jaak. Maybe Rory has to unmute you.
What about now?
Yeah, no, perfect.
Yeah.
Okay. I did not realize I had not muted myself. Okay. Yeah, I have the similar story. I need to give a little bit of background. So I came out of the Estonia when Soviet Union finally inevitably collapsed. I went to study in Helsinki. I was doing my PhD in computer science at Helsinki University with the Esko Ukkonen suffix trees and patterns. All kinds of data mining emerged in there. And then Alvis Brasma joined Helsinki as a visiting researcher, and we were together working on yeast, starting to look for transcription factor binding sites in yeast promoter regions. By the way, I did that on my colleague at Helsinki was Linus Turval, so I did that calculation on his workstation in his office, in his office. It was the Alpha machine. But anyway, I joined the EBI after Alvis had joined. Alvis said that in this industry support group that there is interest in data mining and invited me to apply. So I came April 1st, 1999. And in my mind, the EBI was sort of like still small, but it had already become a center of activities. But with this 120 people, I think, it was relatively small still. But yes, what happened in the Nordics was this huge growth. And I have thought about these kind of three things that I wanted to convey as a message is that what emerged then was actually data science. On one hand, it's all this pattern mining, machine learning, visualization, all this kind of analysis. On the other hand, it was the data that emerged. So 96, I worked on the yeast genome, but 2002 already human genome was available, and that's a huge increase in all kinds of data, sequences, protein structures, maybe not that big as data, but extremely important even now for the AlphaFold, right? So it was emerging of the data and data science methods. But what happened really when I joined, we started to work on gene expression, and I have thought that it's actually the first thing was not about the amount of data, but the first thing was that what Eric mentioned before, that the dynamics or the behavior of the biology. So how do you characterize what is the experiment? How do you characterize what the measurements actually measured? Because in the sequence, you have the sequence, and you don't care which method you get the sequence from, right? But with the gene expression, it was quite different. You needed to have the standards for across the different species, what the biology is. Gene ontology came around. Mike Ashburner had a big, of course, contribution into that. And in gene expression side, we had to define this. What are the minimum biological information that we need to capture in the databases? So these standards emerged about the biology, not only the formats. I don't want to talk about these XMLs and all these formats, but actually how to capture the meaning of biology. And that was a big thing, I think, as well in 2000s. And then Janet is in here. Janet joined EBI as a new fearless leader. I soon left, but not because of Janet, but because I was headhunted back to Estonia. But then there was a realization that EBI cannot do everything for Europe in bioinformatics. All countries need to participate, and all the 2000s went into establishing the Elixir, the bioinformatics infrastructure, which I just checked that 2013 was officially launched, and Estonia was the 5th country member state of Elixir S3 infrastructure. So I was part in that. And that brings me to the third point, that actually that all this, you can be trained at university for certain methods and knowledge, but what the EMBL-EBI gave was this huge network of people, the community of bioinformatics across Europe and world. And this was a huge springboard for later careers. So I would not have been headhunted back to Estonia had there not been EMBL, right? And I came back to actually work on the Estonian Biobank establishment. So this was the reason, right?
Yeah.
And since that, I have had a very, very good career in Estonia. Led the computer science department in here, helped to establish all the biobank, etc. So it's a multitude of these opportunities afterwards. But these 2000s, I think it was all about data science growth, formalizing all the databases and getting the community together in Europe. So that happened in these 2000s, really.
Yeah,
and so that's briefly what I wanted.
I think it's right that what you're saying is that, of course, EMBL can't do everything, but what they definitely have been able to do and can play a great role in is training young scientists. And we saw a great example of that from EMBL Rome, but also helping with some fundamental aspects of data sharing. And Allyson, I know you were, have been heavily involved with the whole concept of fair data sharing. Perhaps you could just share your thoughts about fair sharing.
Well, there you are. That was the perfect intro. I echo what Jaak has said here, because it was the springboard for so many careers, for mine and for so many others that had come to the EBI before and since. And I also think that what you said about the minimal information standards, the reporting guidelines that were starting to come out, for me, the EBI was all about databases. And more than anything else, when I think of the EBI, I think of all the different databases that come from them. And because of that, because it was such a wealth, such a treasure trove of data of all sorts of different types, I think that it was also a place that was a magnet for people who were not just looking for these minimal information guidelines, but thinking about the terminologies and the formats and all of the other ways in which we need to ensure that our data is shared fairly and openly where possible. And from the end of my time at the EBI all the way through to now, I gravitated towards this sort of standardization efforts. In the end, I did my PhD in semantics and ontologies. And as I left, as the last year or so of my time at the EBI, I spent some time with the OBO ontology, this life science ontology, where we were exploring what it meant to describe an experiment. And what sort of words do you need to use to talk about life science experiments? And what does a study mean, and an investigation and an assay? And these all mean different things to different people. And that is really what we learned. And I spoke to you previously, Angus, about how when we were trying to, I say discuss, but it probably got a little more heated than that, the definition of study and the definition of investigation and experiment. You had such polarized opinions, and that has carried forward. That's what science is about. But when you brought together, the EBI was the place where you could bring together all these different minds regarding data sharing. And we had Robert Stevens from Manchester, who was a computer scientist, is a computer scientist, and we had Barry Smith, a philosopher, all shouting maybe is the wrong word, discussing strenuously about the philosophy of ontologies and what makes a philosophical ontology and what makes a computer science ontology. And it was the most amazing series of discussions I witnessed that day. And it really carried me forward. In the end, I went to Oxford to work for Susanna Santasansone, and she used to work at the EBI as well. And that is, in fact, how I managed to get the job with her, in the sense that when she saw my application, she at least recognized my name, and we had coffee together. And it was, as Jaak had said, this giant network of people that starts from a resource like this EMBL outstation. And she is one of the authors, together with Philippe, also an alumnus of the EBI, of the FAIR principles. And just the fact that you can start talking about these aspirational guidelines, Jaak's reporting guidelines and minimal informations, and you can start building up a picture for your particular research domain. What does it mean to be fair, and how far along that spectrum of findability and interoperability and reusability do we need to go for the type of data that we normally produce? It's all fascinating. And I'll stop there because I know we're supposed to keep our answers short.
I will join in about the gene ontology. So I remember one conference presentation where Mike Ashburn was sort of like, I have a dream, kind of, that we have these gene ontologies, and we have experimental data from the gene expression experiments, and then we use gene ontology to understand the experiment, right? And to visualize what these heat maps, how the gene ontology would be there. So I had I was actually working on the software development, algorithm development, and data analytics, data analysis methods. And then I went and implemented the proof of concept for this kind of enriching gene sets with the gene ontology terms that are over represented, and that was at the EBI 2001 or so. So one of the first implementations of this sort. Actually, coming back to Estonia, we made a better version of that tool set, GProfiler, and that has been heavily used, I think, with nearly 10,000 citations altogether by now. How people are actually using this kind of line of thought, how to understand the experiment. But it was these different components together. And about the EBI, when I was what was in the previous discussion, that you shouldn't limit your research into one question only. I mentioned this pattern discovery in the DNA, the gene regulation aspect. And then a couple of guys from the EBI came to me that, oh, this tool of yours, can you work with the protein sequences? Of course I can. It's just from 4 letters alphabet I went to 20 and no problem. And then we went on and actually solved, first time showed that for G protein coupled receptors, their coupling specificity type. And that was also really nice, that using the same similar tools that you can cross from one biological domain to the other. And the mix of these people who ask different questions, work on different data. But bioinformatics is really nice because you can jump these very specific questions from one question to the other. It was hard to understand back then because people had, oh, what is the biological one question that you're working on? But in my mind, I think it was important that we can have methods that can have multiple applications.
Yeah, so Jaak, I actually had a question about that. I mean, I think an interesting aspect there is that, of course, this was the initial time in which we were confronted with all these mass of data, right? I mean, usually before it was like, one experiment, a small data set, and now you had like tools to produce so much data. And I mean, you refer to these, and you guys both refer to these in building ontologies, which essentially means building categories, like building a universe. And this is somewhat, almost like, beans in which you put your data to organize them. So this is like an act of creation. And Allyson mentioned a philosopher. I mean, did you find out that you were building, beans that were not filled with data, so that then became useful later on?
I was standing on the shoulders of giants, so I was using ontologies. I was showing how the ontology could actually be used in that setting, and I was not directly involved in defining, because I did not know that deeply the biology itself. But I was using the results. I was using the gene ontology of that time, and I was also building all the tools for gene expression data analysis. Actually, we came often with Alvis. We came often to Heidelberg into these different courses that were about the gene expression data analysis. So we were teaching there the data analysis methodologies, how the clustering works, explaining all of this. So I was myself working on the tools and algorithms rather than building the ontology. But this is the beauty, that you have people who create databases, maintain databases, who create this semantical understanding, what should be there at our Express group. So Alvis was promoted to group leader, so I became the first member of the Express group back then of his. And when I left EBI, I left just before we started to admit the first data sets. But I was working on the gene expression data analysis software at that time. So,
sorry.
Yeah.
I wanted to bring in also Allyson, because maybe she wanted to pitch in on this.
I just wanted to shout out to the Go Girls. I don't know if you remember, Jaak, they called all the gene ontology annotators, they call themselves the Go Girls, the Midori and all of the rest. Yeah. And I think I saw Jennifer here on the list. She's Interpro, but she had, we were all dealing with the Go annotation for years. It was as it was getting off the ground. And I know I saw Bob here as well. A few of us from the EBI from that era were there, and it was just absolutely amazing. They were for a very long time only women who were building gene ontology from the start. And I just remember seeing them work. Yes, I started working on EBI, but I wasn't working to the same depth that those women were building the gene ontology, which has become such a massive thing. And I know Michael Ashburner definitely was the founding sort of father of it. And I remember him coming into the EBI, and he was such a big presence, his big beard, his white hair, and he would come in and he would sort of Brian blessed the room. I sat in the same room as Eleanor, who was in Michael Ashburner's lab before she came to be a curator at SwissProt. And she and I were friends while we were, the whole time we were there. And he would always come specifically to see Eleanor. So he'd wander into the room, he's like, hello. And then he'd sort of come over to Eleanor and have a nice chat with her about flies. And then after talking about flies, he'd sort of wander out, and it was amazing. I mean, you don't realize at the time
He was dominating big conferences as well from the stage.
Well,
Yes, yeah, he's a personality. He was certainly a personality.
I think we still have Janet Thornton in the audience, and I wonder if, I don't want to put her on the spot, but Janet, you've always been one of my scientific heroes. I wondered if you might like to just share your memories of trying to oversee this huge explosion in bioinformatics in the first in this decade, in the first 10 years of the 20.
But Angus, we had such good people there, like Jaak and Allyson and the Go Girls, and of course Michael as well. And it really was just a case of letting people get on with it, I think. I mean, sometimes I look back and think, I didn't do anything at all. I just made sure people got on with it and enjoyed it. But I do remember just an anecdote about Michael, who, as Allyson said, was this huge, he used to wander in and usually at that stage, smoking his cigarette. And he told me about the Go annotation. It must have been very, very early on. And I was just staggered. I really did not think it would work because it was so complicated, so huge an endeavor in my mind. But Michael was very canny about the way that he did it, because he very much got the databases, in particular UniProt, on side. And he worked with Rolf, and they came up with this ontology that actually very, very quickly took over, because there wasn't anything else. There really wasn't anything else. But developing the concept, as Thomas said, developing this idea of bins, what are the bins? What's the hierarchy? Is really quite an intellectual challenge. And I was amazed that Michael suggested this, because I thought it was so infinitely difficult. What I didn't appreciate, of course, was that he had, at least at that point, 40 years of experience in biology, mostly in Drosophila, but actually he understood about biology, which I was still and am still learning. It's quite fascinating. There are so many different aspects. But because Michael had been working on the flies for so long, he knew all the different aspects of their biology, and worked also very closely with other people. And he was very good at that, at rallying the troops, etc. But as you say, and Kath Brooks-Banks, who's been head of our training at EBI for so long, she started out as a go-go. And, it was a great time, Angus. Yes.
Yeah, no, absolutely. And thank you for that. So I don't put you on the spot, but since you're here, you saw this, oversaw this fantastic development of the EBI and the central role that it's taken up in pervading the science that all of us do. So actually, we're all computational biologists in one way or another, because whatever happened. Perhaps you could also share your thoughts, Janet. Where are we going in the next 10 years?
Janet made this elixir happen, really. That was also a huge contribution.
Oh, it nearly killed me, elixir, but that's another matter. In terms of, so it's interesting, because when I joined the EBI in 2001, so just after Jaak joined. And of course, that was the year of the human genome. And I don't know whether I foresaw, but to me, it was absolutely obvious, and really has been ever since I've worked in computational biology, that this is the essence. Maybe it's the essence, because what we're always trying to do in biology is find models to understand what we observe. And it's exactly the same in physics, with astronomy, finding models for galaxies, but we're finding models for life's processes. And the only way to do this is computationally. So, I mean, Jaak has been involved with integrating lots of data from different aspects, different data types. And, I soon discovered that, in my brain, feedback loops and those things are so difficult to understand. And the only way to do it is by simulation and computation. And Eric, well, I can remember when you were in Heidelberg and doing all the stuff with the spindles, etc. And it's the only way that we can sort of capture the essence. And so going forward, I think this will be, computational biology will continue to grow. And as you said, Angus, everybody is a computational biologist now, because you can't do the work without accessing the data that underlies it. But to me, in the next 10 years, the major areas will be the advancing more into the medicine and more into the environmental and the biodiversity aspects, and really also understanding evolution. We understand it so much better than when, Angus, you and I started out. The molecular evolution wasn't really hardly there. We know so much more, but I still feel that we've hardly started on that. And its application, the application of AI, obviously, but just the data, gathering the data, I think will have a huge impact on medicine that will affect possibly even my life. I'm not sure I'll be around that long, but certainly for the younger people, their lives. And the biodiversity is just fascinating, and we can really capture all the organisms in either in seawater or in soil just by doing the sequences. Isn't it fantastic?
Inspiring.
We can do.
My research group at the moment, we are entirely working with the electronic health records, so trying to understand biology from somehow to take these very fragmented, sparse bits and pieces about the health data. So at the moment, we are even not working on the molecular side, but on the health data, but eventually they will come together again because of the personalized medicine. Yeah.
I think it's very interesting, isn't it? I think you guys are absolutely right. I would add an extra thing that I'd be looking for in the future, and that is more. We've always had, especially at the EBI, a real view, and EMBL as a whole, of cross-disciplinary science. But I think a lot of that is really going to increase. In my mind, it has already done so. I'm biased and influenced by the career that I chose, but it's very much a case of when we just wrapping up a grant, this BioCovid grant, which the EBI is a part of, and Henning and a variety of people from within the EBI were working with us on that. And that's BioCovid was for standing sort of, not ideally as an acronym for beyond COVID, looking at how we would share data in the next crisis situation around infectious diseases. And the data that we were integrating wasn't just biological. There were people from the social sciences and the various EOSC and the various social sciences and humanities infrastructures within Europe. And so I think that it's not just for me about building strategies and minimal guidelines and reporting standards and repositories that focus just on one research area, but it's about bringing data stewardship and good data management across these subject boundaries, and sort of seeing what everyone's solutions are to try to move forward into sort of high quality data sharing. When I talk about it, I talk about it like I'm trying to convince scientists to be like good preschoolers or kindergartners, and how you learn to share in when you're 6 and 5, and how where it's not sensitive, where that makes so much sense as scientists, and trying to let them, let go of their research data for the common good and let it fly on its own.
So maybe one final aspect that I think is worth mentioning is that I found very fascinating is this aspect of the fair data being able to be understood by the machine on one side and humans on the other side, which is becoming increasingly difficult. And perhaps, one final question that I had maybe for Janet was that this was one of the times in which you also had to convince the EU, or anyway the EMBL financiers, that there is an infrastructure to build, right? European-wide infrastructure, because now you have the data, you have to move the data, you have to exchange the data and everything. So how challenging was that? Because I see that every time, an infrastructure needs to be built, every country says, I'm not going to put money into this. Is it really worth it? How was it?
So I think it was the most difficult thing I was ever involved with, without a shadow of a doubt. And I used to say to Ian Mattei, was DG at that time, and I can remember saying to him, it's easy for you, Ian. You've already got EMBL set up. You've just got to run it. But actually trying to create something like Elixir that involves signatures from the member states and bring people in, well, it was difficult, but it was also totally fascinating to see how it evolved, because it didn't turn out as, in many ways, as I'd anticipated. I learned a lot about how you can try to bring countries together. I really don't envy politicians and diplomacy. I mean, it's hard work and it's exhausting, because you have to go around and see everybody. But seeing how science was organized in all these different European countries was quite revealing, because it is actually, or was then, quite different. I think there is a convergence to some extent compared with how it was. But even so, even now, the organization of science in different countries is different. But I still believe in it. I mean, it sounds silly, it's sort of, but the importance of creating something like Elixir, where all the member states can join together and work together for fair data, for sharing data, for developing infrastructures, for not duplicating the effort, to me is a really important thing to keep at the forefront of our minds. And I think how the medical data bit, which is totally fascinating, but that's sort of, it's part of Elixir, but it's not part of Elixir. I mean, it's a complicated boundary between these two. But if that boundary has to be crossed again and again for us to understand diseases. But actually, I don't know if there's time, but since Eric and Nadia are on the line, I would like to ask them the question that you asked me. How do they see the future in the next 10 years?
Could we come back to that in just a second, Janet?
Yeah,
because there's a bit of work here. Thomas and I work behind the scenes,
right.
and we'll come back to that, I promise, in just a second. But we had just lined up Matthias Haury as well, who's been patiently waiting, and especially, so we've been on this journey where we started with Eric in Heidelberg, and then went to Nadia in Rome, and then to all of you at the EBI. But this aspect that's come up of training is so central to one of the biggest things that I think you should all be so proud of, the contribution that EMBL's made. And perhaps Matthias, you were involved in the whole training program, I guess, at EMBL.
Yeah, it was a very interesting time. I joined about mid 2006, when things were already just getting started. The ATC was just in the plans at the time. And I came from a journey mainly abroad. I'm actually born very close to Heidelberg, but never really interacted with EMBL other than, I don't know, many years later, because I lived in France and US and Portugal before. But I think this international aspect of EMBL really fascinated me, and that was one of the reasons why I joined, not knowing that an ATC was actually in the plans. But I think the fact that EMBL made it possible to do so many innovative and new things, especially like working with Eric, with Janet on trying to build new programs. The outreach aspect was very important, but also the fact of trying to do new things. I don't know if many of you remember what iPods actually stand for, but at the time we're like with Detlef Arendt together kind of created this EMBL interdisciplinary postdoc program, which I think still exists and was a very innovative idea at the time. I think it has been extended to the PhD program as well now. So I'm really amazed, and I still think the stage setting that EMBL does on the training across the member states and even across the world is really amazing, and I think it's a unique place. I still, even being now back in the US and seeing it from the other side of the ocean, I think it has that enormous impact that I think we can all, everybody that worked on it can be super proud. And I think the one other aspect that I really appreciated and enjoyed is the fact that people understand that science has to reach out to society. And I think Eric is a big proponent of that, of course. But I think even with Rosanna and Nadia in Monte we've done lots of stuff. Science in school was a big effort, like getting back to the teachers and engaging the high school students and students early into the science career. I think is a very important point, and I still up to today think that this is one of the aspects to ensure that the next generation of scientists continues to become scientists and not end up somewhere else in business all over, are really important aspects. So that EICAT training program, which I became coordinating manager, which is I think about the longest job title I ever had in my entire life. But really, with the building of the training center and the build-up of the course and conference program, enabled so many things. It had one interesting challenge, and I think you all mentioned that earlier in the talk, is to get now the increasing presence in different member states of entities, like Monte in their EBI coming on. I think there was talks about Barcelona at the time. The outstations in Grenoble and Hamburg, how to bring all of those in and keep collaborating and find ways to kind of communicate and connect them. I think especially also for the PhD students was an interesting challenge. And I found it really fascinating how flexible EMBL was and still hopefully is to kind of go new ways and try new initiatives and new ideas. And so one of them, for example, that, okay, might sound silly today, but we're all sitting on Zoom today. At the time, we had this idea to kind of create this very interesting Nobel laureate lecture series for the inauguration of the ATC. But I wanted to kind of have the member state participate. So to be able to stream lectures to different countries at the time was completely unheard of. And we really had to even set up our own infrastructure, like QuickTime streaming servers and hack stuff together to be able to do that. But that just shows how enabling EMBL actually really is, and how many things you can do that you probably wouldn't be able to do anywhere else.
So Matthias, I was actually curious because now you move to the other side of the world and you are part of a new institute, right? Which I find really fascinating, which is the Chan-Zuckerberg Institute. And so I was wondering, I mean, what happens there in terms of what you're doing and how much, the experience at the EMBL and has, changed or instructed the way you are dealing with things there?
Well, the world is very small. I think when Nadia was talking about Peter Cruz, giving her advice, when I moved over to Florida and started to kind of help building the Max Planck Florida Institute for Neuroscience, Peter Cruz was the chairman of the board. And I had him actually known him from a Boehringer Ingelheim fellowship that I had during my PhD in Paris, way years earlier. And now moving to the Chan-Zuckerberg Initiative, we're building up a new imaging institute, but interestingly, of course, a corporate partnership program that we started at EMBL with Zeiss and Leica and many of these companies at my time ended up kind of maybe facilitating the creation of this fantastic imaging center that you have now there. Now, of course, we on our side here, the most important thing we want is to collaborate with EMBL again. So the circle closes itself now after many years, and I'm actually back collaborating with Jan and Julia and people over there, trying to kind of build this bridge across the Atlantic now. And even with the EBI, realizing how important data, like structured data in the imaging world is for AI in the future, makes it clear that we have to collaborate on a worldwide basis. And I think this is the fun part and the really exciting thing that we can do this now. Even also thanks to all these alumni relations that we keep alive. I think it's really fantastic.
Yeah, I mean, it's fascinating because if you think of it, I mean, imaging data are, of course, data, and there's a lot of computational analysis that you can do, but somehow is the next level of complications, right? Because it's much more complicated than raw genomic data or quantities. Yeah, right. So it's a new challenge ahead. Well, actually, the other thing that has really nothing to do with this, but I wanted to ask is that of course we know of the program at the EMBL, the EICAT, also for the big new building, which is a bit shocking, especially for people like me that live at the EMBL without that, and then you see the big helix coming up. So can you tell us a little bit of how that came about? I mean, it's such a fascinating building, right? And I feel that buildings are also part of what then starts inhabiting a building, right? So if you have a beautiful building and then you have a noble challenge, then you are even better off. So how was about the building there?
Well, the funny thing is, when I got hired, nobody told me about the building until I signed, and then after I signed, I heard about this big project of the ATC, and yeah, it was very interesting because I think probably all of you know that Klaus Tschira from SAP and the Tschira Foundation had committed, I think, 10 million euros to kind of build that building. He brought the architect with him, and the architect was somebody that was, of course, a bit challenged by the fact that this was supposed to be a double helix, and previously he was used to building square buildings, and so the whole aspects of trying to build something round, and then there were requirements, for example, for evacuation in a fast manner. So that I think those of you who remember, the ATC has this ramp outside of concrete that had to have a certain inclination so that people in wheelchairs can actually still evacuate safely or move up and down. And so the architect had this idea that these whole ramps outside should have been precast somewhere in Germany and then being lifted up to the hill with a cargo lifter, which was at the time some kind of a startup with a Zeppelin, ex-Zeppelin, big air balloon kind of things that are supposed to carry huge amounts of weight up the hill. But by the time that we started, actually, that company had gone bankrupt. So then one had to figure out how to do the casting of those ramps on the building itself. And this is interesting challenges because concrete kind of settles, and even I think if you go to the roof terrace, the roof is also kind of an inclined roof, and that roof settled by more than 20 centimeters down in as part of the solidification, and all of that has to be calculated. So there were a lot of interesting challenges on the construction side to solve, which of course we had to go through. Now after that the building is there and standing is beautiful and really, I think, very functional. Seems trivial, but in the days was actually lots of discussions, and there were interruptions in the construction process for a while because there were changes in the EU rules of how much steel one has to put into concrete pillars, and suddenly the steel we were supposed to put in there was way more expensive than we had budgeted. So there was questions of, okay, how can that be financed? Very interesting little details, but it was an exciting time. And then being able to open that and run it and build this course and conference operations that I think now is unparalleled worldwide. I think the courses and the conference that EMBL runs are fantastic. Maybe one little anecdote to that. I remember early on we were with Jennifer Liebigon-Schwartz and Jan and Matthias and Anne trying to figure out, okay, what's the first or one of the first inaugural symposiums? And I'm an imaging person, and so I was really excited about this imaging symposia. We created the EMBL symposia. There were lots of discussions whether it's EMBL or EMBL, but in the end turned out to be EMBL. And then the question is, how do you kind of market that? How do you kind of get that out in the into the science community? And we came up with this title, Seeing is Believing. And there were lots of discussions. Okay, as a science institute, we actually put a title that believes in something, or do we really kind of want to work with facts? But I think history showed that was one of the most inspiring titles, I think, for a conference, because that conference is still around and is still probably one of the biggest imaging conferences in the world right now.
Yeah, and I mean, if I have to add, I think it applies also very well to the museum, the imaging museum that is now part of the imaging facility, because it really happens. Seeing is believing.
Yeah. I mean, although from some of our previous alumni chats and seeing what's happening with AI generated images, maybe seeing shouldn't be believing in the future. So this would get us back to the question I unfairly threw at Janet, and she was asking. So Matthias, if you'll stay, and maybe Eric and Allyson and Jaak and the other speakers, we're into the last 10 minutes. I think just as we expected, the time has flown by. We've really loved the discussion. But please share with us, I guess we've got 10 minutes left. So maybe if you each could just share with us a couple of sentences. Where do you see EMBL and the future going? We've just been talking about where EMBL was, 15 years ago and 20 years ago. Life has changed so much. What do you forecast is going to change in 10 years in the future? Eric.
To me, I think that the problem of evolution. I mean, I think that evolution is a big problem that has been barely addressed, and what we found barely addressed at the level at which it should be. Interoceans, for example, we discovered like in the ocean there is something like 150 million genes, or probably more than that. I mean, a huge number of genes. And when you look at this, at the role of interaction between viruses, their hosts, the transfer of genes, all these things. And I think one of the big questions is how life became so complex over evolutionary times. Everybody talks about selection. Selection is interesting, but not so much. I mean, what is really interesting is the origin of complexity. And, how genes got, how the diversity and complexity of genes that have been generated, how this has been created probably in the ocean first and then on land in other things. So I think this is a very important thing. Another aspect which I think is very important is, when in cell biology, when we started the use of computer simulation to try to understand, as Janet said, we try to understand the organization of the spindle. I think that most of the organs and structures in the cells are out of thermodynamic equilibrium. They're dynamic. And we still don't understand very well how all this works. So I think that there is a need for a lot of work on the development of computer science, but not only in the sense of analyzing data, the large data sets, but also trying to understand how interaction between many different types of molecules or organisms generate complex systems also. On one hand, you have the problem of evolution, how complexity emerged, and how this complexity of this molecular complexity can generate very, very important structures now. I mean, to me, this is what I see for the future.
Okay, Nadia, what's your, in a nutshell, what's your forecast for the future?
Well, I mean, I have followed in Janet's footsteps, as I have done all my life since I've met her. And when she says that really the next 10 years is going to be finding models for life processes, I think I've taken that to heart. One of the things that's very clear to me, having worked in the mouse for so long, is that really the connection between organisms, as Eric says, has to be looked at not just from an evolutionary point of view. Obviously, we've lost our ears and our tails, but more from a systemic point of view, looking at, the sort of the population genetics and how the systems of genetics play with the environment. And another thing that has really come up in the last 10 years, and I think is going to really predominate, at least in my field of modeling life processes, is thinking about how we've sort of almost sidelined the whole topic of physiology, which really is a very old word for systems biology, but it really does actually matter how different processes within a body work together. And in that sense, I think what we're going to be able to do with the extraordinary explosion of deep learning is to find patterns, not only just in the physiology of organisms, but in the cells that are derived from those organisms, where Eric's whole life has been really focused. And to me, this idea of self-organization as a kind of counterpart to evolution is something that we simply can't ignore. I just read a review of Richard Dawkins' new books, and he's still ignoring it. And I'm still hoping that before he leaves us all, he'll just tip his hat to the fact that there are proteins that interact with each other. There are systems that interact with each other. And I hope that that's where we're going to be illuminated the next decade.
Great. Paschalis?
Yes, I think I will leave you with a book that I'm reading about Jennifer Doudna, The Code Breaker, where basically I think we have become one of the many species, if you want, like bacteria and viruses, that we have now the ability very easily to engineer our own genome, as bacteria and viruses do for their own defense. So the big question for me is whether how far CRISPR can take us to cure genetic diseases, and there's a lot of bioethics with that. So far, CRISPR has been very effective on engineering somatic mutations, right, or correcting mutations that are not transmitted to the next generation. But obviously, there is a big ethical discussion on whether it's correct to cross the germline, right, and then having these mutations for the next generation. So I think genetics will impact in a profound way the life of patients, and there will be a profound discussion also in bioethics.
Okay, we've only a few minutes left. Jaak, one sentence, where do you see the future going?
I think what is important for EMBL is to find some balance in between. I'm not trying to prescribe any specific research question, but balance between being this big center, equipment, data, and finding something where all these people are inspired to work on the problems that no single university can tackle so easily. So there has to be something that is inspiring about EMBL for future. Really, and this is really important. Speaking as a council member of EMBL, I think it's really important that EMBL is able to find that something uniqueness.
Yeah, I think it has to be inspiration. I 100% agree. Allyson, one sentence, future?
Oh, elevator pitch. I love it. I think it's not just fair. I think looking beyond fair, a fair outside of data. So there's already papers published of fair in software, fair in training materials, and all sorts, but more importantly into care, and cares around equality, diversity, inclusion, justice, ethical data sharing. And that touches upon the biodiversity field that we talked about earlier and that Janet mentioned was a big thing coming up. And that is where I think we will find some really interesting stuff happening.
Matthias?
Yeah, I think I'm just summing this up. I think both what Eric and Nadia and everybody says, I think the strength of EMBL will be to be able to combine both the data and the AI and ML side with the experimental side. And I think to have these two communities talk and feedback and loop with each other, I think is super important. And EMBL is in a very unique position to be able to do this on the European side. So I'd be really hoping that will be the one thing that EMBL will stand out in the future.
That's great. Janet, if we leave you with the last word, was your question answered? Or perhaps we need to dedicate a future episode of Coffee with EMBL to this amazing topic?
I think so, because it's interesting, because we all come from such different perspectives, and hearing everybody's, well, in fact, everybody online, not just people all think differently, and it's just great to hear and synthesize it all into what might happen in the future.
Yeah, I think I agree with all of you. I also want to give a quick shout out to Nadia and the EMBL Rome, who I believe are having a celebration coming up very shortly. And I hope the celebration is fabulous, and your dancing and celebrating is in the true EMBL style and does EMBL proud. I would also like just to send a reminder to everyone. Please do check out the previous alumni chats that are recorded and online. This one will hopefully be also available online very soon. And also the ongoing series. If you enjoyed hearing from Eric, you should be able to see, if not already, very shortly, an extended interview with Eric that we recorded earlier this summer. for the YouTube channel that we're creating. And Matthias, this is maybe in the spirit of what you were forecasting back then, that we've created a YouTube channel and we're recording interviews and we're looking for more volunteers. Janet has kindly already conducted an interview, Eric has, and we have a list of interesting people, some of whom are online. Mary Holmes did a fantastic interview with Thomas, and please do check those out, volunteer to take part, and do give us your suggestions for future topics for these alumni chats. We've just had our 30th alumni chat, and again, I'm overwhelmed that we've done 30. I hope we'll do many more and continue to bring the community together. I think just to leave us all with the last word, that what I take away from this is inspiration. And I think the future of EMBL has to be inspiration. And as we started off with Eric saying, believing that everything is possible. I think that was just such a fantastic quote. I want to put that up in my lab and just say, believe everything is possible, and then inspire, inspire people to go forward and make it possible. I'm absolutely thrilled to have had the privilege of talking with you all today. Thank you so much to all the participants, and thank you to everyone who joined online. We really appreciate your support. And we look forward to seeing you at the next Coffee with EMBL.